Thank you so much. You may be seated.
Ladies and gentlemen, colleagues, students, family and friends, good evening and welcome to this inaugural lecture in the Faculty of Science. On behalf of Stellenbosch University, the Faculty of Science and the Department of Botany and Zoology, it's my privilege to welcome you to this important academic occasion. I extend a particular welcome to the members of the Rectorate, colleagues from the faculty and the wider university, collaborators, postgraduate students, and all guests joining us this evening, both in person, and I believe there's some online as well. A special welcome is also extended to Professor Tammy Robinson-Smythe, and your family, your friends, your colleagues, your students, and your collaborators. I would also want to especially welcome her husband, Wynand, her two children, Damien and Kira, her mother and father, Derek and Sharon, as well as her godmother and godfather, Jill and Mike, all the way from Carltonville, and they nearly fell into a problem with water when they travelled here. So welcome, and we're glad you're here. Inaugural lectures are formal university occasions, but they are also deeply personal moments. They mark an academic milestone, but they also acknowledge the people who supported, encouraged, and walked along the professor, whose work we are to celebrate this evening. The inaugural lecture is one of the important traditions of the university life. It gives a new appointed professor the opportunity to present the intellectual journey that has shaped their scholarships and to set out the questions that will guide the next phase of their academic contributions. It is also a moment for the university to pause and to recognize excellence, not only in research, but also in supervision, in mentorships, in collaboration, but also service to society. Tonight we have the privilege to hear from Prof. Tammy Robinson-Smythe, Professor in the Department of Botany and Zoology, core team member of the Center of Invasion Biology and holder of the Research Chair in Managing Invasions and Protected Areas. Her work is situated in a field that is scientifically complex, but also of immediate relevance to society. How we understand, manage and respond to biological invasions, particularly in marine and protected environments, are important to us. Professor Robinson-Smythe's academic background is in marine ecology, and I believe from Port Elizabeth University, as was told to me earlier. And her research was focused on the spread, impact, and management of invasions. She has a particular interest in the interface between climate change and biological invasions, a nexus that is becoming increasingly important in ecosystems placed under growing pressure from environmental change, human activity, and shifting patterns in species movement. Her research group, based in the Center of Invasion Biology at our university, works on marine alien species along South Africa's coastline. Her group combines field surveys, experimental ecology, and management-focused approaches in support of national efforts to safeguard marine biodiversity. Much of this work is focused on marine protected areas, where the need to balance conservation, management, and evidence-based decision-making is of particular importance. This is precisely the kind of science that matters to us. It is rigorous, but is not removed from the world. It asks fundamental questions about ecology and change, but it also speaks directly to conservation practice, policy, management, and the long-term protection of our natural systems. Prof. Robinson-Smythe's work also reminds us that excellence in science is often a team science. It depends on postgraduate students, postdoctoral fellows, collaborators, field teams, managers, institutions, and communities. It requires careful observation, strong methods, persistence, and the ability to work across disciplines and institutional boundaries. In this respect, her work reflects one of the strengths of the Faculty of Science, the ability to produce knowledge that is both academically robust and socially relevant. Her profile also reflects strong academic leadership. She's a co-editor-in-chief of New Biota, an international journal Invasion Science, and is a core team member of the Center of Invasion Biology. She's also contributed to the broader professional service in the field, including editorial work connected to invasion biology and protected area science. Inaugural lectures in our faculty remind us that these evenings are not about listing achievements. They're about introducing the person, the scholar, the wife, the mother, the child, and the academic journey behind this work. In the same spirit, tonight is an opportunity for us to recognize Professor Robinson-Smythe, not only for her research outputs, but also for the intellectual direction she brings to this important field, the students, the young researchers she is helping to shape, and the contributions she continues to make to our wonderful university. Tammy, tonight is an important milestone. It is a recognition of the work that you have done, but also the work that we are hoping to look at and see what will come. Your research speaks to some of the most pressing ecological questions of our time. It also reminds us why universities matter, because they create the space for us to ask questions, to build evidence carefully, and to contribute meaningfully to society. Above all, you are a woman in science. As a fellow woman in science, I wish you personally a good congratulations to you, to your family, and I hope that we will just see how you strive and become better in what you do. I know how difficult it is to have a family, a husband, 2 children to look after, school runs to do, and still to read papers and produce work in the evenings. Thank you. Ladies and gentlemen, it's now my privilege to invite the department head, that is Professor Konrad Mathieu, to give a short little introduction and also congratulations. And after that, Professor Tammy Robinson-Smythe will give us her inaugural lecture. So please join me to welcome Konrad and then afterwards Tammy.
Thank you very much. Tammy is the most important person here this evening. It's indeed an honor and a privilege to share this moment with you. I think I want to celebrate Tammy's career as an academic in our environment who combines scientific excellence, practical conservation impact, international leadership, and genuine mentorship as her flagships. So, as was indicated previously, or by our previous speaker, Tammy's history started at UPE, which is now NMU, was in MMU in between, and there she was actually the best final year student in zoology, got the prize. Then she moved to UCT, where she did a PhD under 2 A-rated scientists, George Branch and Charles Griffith, and another person. But Tammy, I think, and there you also won a prize for the best PhD. And I think that excellence that you display throughout your whole career is evidence. What's striking about you is that you never separate scientific excellence from real-world relevance. You're one of the leading voices in marine bioinvasion, as we've heard, and you've published close to 100 peer-reviewed papers, and you cited 700 times last year alone, which is quite nice. So you didn't know that I checked. Okay. Statistics alone does not tell the story, of course. You helped to lead the first marine eradication program for the invasive species in Africa. Your work influenced far beyond South Africa. You collaborate with scientists and agencies across 6 continents. I think that's all of them. and you contribute to the international assessment, including the UN World Ocean Assessment. And in 2024, you became, as we've heard, co-editor-in-chief in the highly respected journal Neobiota. But at Stellenbosch University and within our department, you really build an extraordinary legacy of mentorship. And you've supervised more than 20 MSc and PhD students, more than 30 honor students who are not listed in your CV, but very good. And many of these students actually build strong careers. And I think part of that is one of the most important things of being a professor is not only the knowledge that you create, but also the people that you inspire and develop in that process. So thank you very much for that. And what stands out is the balance that you bring. Tammy is rigorous, never nasty, and you are highly productive, while you still always think about your students and how to collaborate with your fellow staff. Thank you for that as well. You are a research chair in managing the invasions, invited to several keynote speakers, as a keynote to give keynote speeches at conferences. Your editorial leadership, your growing international recognition, all of these point to the same thing. A scientist at the forefront of their academic career, where you are right now. And yet, we know you not only for these achievements, we know you for your generosity, collegiality, calm leadership, and willingness to always support others. It is therefore a great pleasure to also say that I'm very happy that I sat in a committee years ago with a very difficult task. Are we going to appoint Tammy Robinson because she stood against very strong candidates? And I'm so glad we did. So, Tammy, we look forward to hear what you have to say, and all the best, and well done. We're very happy to share it with you.
Right, hi everybody. Sure, I think it's easier to present at international conferences than it is to do this. Thank you very much, Rija. Thank you, Conrad. It's a great pleasure for me to take you through a little bit of a whirlwind of the work that I've been doing and that speaks to marine invasions in South Africa. I think it's a really wonderful opportunity to share with you the work within this field and what we see coming in the future. So I thought we'd start right at the beginning. Like, what are alien species? We need to know the building blocks that we're working with here. And essentially, what we're talking about when we refer to alien species are species that are in areas where they don't belong. So if we imagine a species that comes from the Mediterranean Sea, and we see it along the Cape Peninsula, there's been an entire Atlantic Ocean between those 2 points. But you can see by those photos that those two areas look very similar. The environments are actually very similar. And the only reason that those species don't occur naturally in both areas is because they can't get there. So when we move organisms across those biogeographic boundaries, we create the phenomenon that is an alien species. But how exactly do we move them around? Primarily, we move them in relation to shipping. So hull fouling is what we're having a look at here. That's a very large ship that's being pulled out. It's going to have its hull cleaned, and all of the brown that you can see on it is fouling. And you can see some organisms are beautiful, and you can see the beautiful orange ones down at the bottom. And as those vessels move around in the ocean, they move around all the animals and the plants that are growing on the bottom of them. Things are also moved around in ballast water, and here you can see a ship with water that seems to be pouring out of it. That's ballast water that it's taken on board because it didn't have any cargo. And ships that are in the state that that ship is in are actually quite unstable. It's sitting very high in the water. If a storm, like what we had earlier this week, hit that ship, it would have a problem. So if they don't have cargo, they take water on, that fills up the vessel, makes it heavy, and it sinks down into the water, and then we wouldn't see where the paint color changes. That point is called the plinth line. And if the plinth line is at the waterline, that ship is the most stable that it can be, which means it's the safest it can be. The problem with ballast water is when you take that up and you move to another place in the world and you pump it out, you're pumping out all the organisms that are in that water. And you know that there's a lot of ships that move around the world. So a lot of things are being moved by ballast water all of the time. To a lesser extent, we move things in terms of mariculture. So these are organisms that we move so that we can grow them in particular places. Here you can see oyster cages from Saldanha Bay. Those envelopes that you're seeing have got Japanese oysters in them. And we grow them here because lots of people like to eat them, and we don't want to be importing them. And then one of the less important ways things are moved around are the pet trade. But this is becoming increasingly important because it's very easy to order things online these days. So we're seeing an uptick in the importance of the pet trade. But the question is really, why do we care? Why do we care if things move around? Well, we care because they have impacts. And they have a variety of impacts. Firstly, ecological impacts. Here we can see a picture of the Mediterranean mussel, and this is actually in False Bay. This is a mussel that does not belong along our shores. And back in the 1970s, if you'd had a look at the shores of False Bay, you would have seen large grazing limpets the size of my palm. You don't see those anymore. What you see are mussel beds. And that's changed the entire system. It's changed from grazers to filter feeders. It's changed the food web. We also have economic impact. So here I've got the same photo as what I had before, because there are multiple economic impacts associated with alien species. The oysters that are being grown bring economic input, bring job creation, and economic benefit to the communities involved. But all of the brown that you see growing over the oyster cages, that's actually an alien species as well. And that's a hydroid that smothers those cages, reduces the water flow to the oysters, and so causes economic loss to the farmers. Here we see a fishing vessel, luckily not along the South African coast, but in Japan, and they had been intending on hauling out fish, and what they hauled out instead were jellyfish. So you can have whole losses of fisheries due to alien species. There's social impacts. This particular photo was taken in Milnerton Lagoon, and those are all reefs that are formed by tube-building polychaete worms. This area was a hotspot for canoeing and kayaking. These reefs have become so big that it's actually closed off, and the canoe club can't use that area anymore. When the wooden bridge that you can see in the background was knocked down, some of those reefs were removed unintentionally. And so a little bit of that use, that recreational use, has been opened up again. And then there's health impacts. And here's an obvious one. It's a jellyfish, but we can have organisms that release chemicals into the air that can cause respiratory distress. So there's actually quite a big variety of health impacts that alien species can cause. Right, let's freeze alien species for a bit and think about the South African coast. It's absolutely a unique place. If we look up the east coast, so KZN, we're looking at really warm tropical waters. We have the warm Agulhas Current. Let's see if I can turn this on. There we go. There we go. So we've got the warm Agulhas Current moving down. It comes off over here at the continental shelf. And then we've got the cool Benguela Current that moves up the west coast. So we have a very cool, temperate region on this side and a very different tropical warm system on that side. And then right in the middle, we've got the south coast or the Agulhas region, which is that nice mix in between. What this results in is very different systems. We've got coral systems with incredibly high diversity, mangrove systems, beautiful high diversity on the east coast. On the west coast, we've got lower diversity, but we've got much higher productivity. So the water is green in this beautiful kelp picture here because of phytoplankton bloom. And that forms the basis of a really important food chain, which results in lots of food production on our west coast. So most of our fisheries are based on the west coast. On the East Coast, we derive other benefits from our coast, recreational, people who fish for subsistence fishing. So we gain food and ecosystem services the whole way around this coast. It's very unique. There's not another country in the world that has a cold system, a warm system, and a mix in between. And because of all of that uniqueness, we have developed one of the best marine protected areas networks in the world. We have a whole lot of MPAs which are tight along our coast, but we also have important MPAs offshore which are protecting cold coral reefs, deep sea systems, a lot of regions that are important for supporting fisheries, but also regions which might come under pressure from oil and gas exploration in the future. So we're now understanding this really important role that MPAs can play. And it's interesting because they have a few primary aims or imperatives. The first is to protect the ecological integrity of the area. So we're looking at protecting biodiversity and the ecosystems that function within those areas. They have a mandate to protect economic interests. So there's a lot of commercially very important fish that breed, and the little ones actually spend time in coastal areas and in estuaries. And if we don't look after those areas and we allow fishing in those areas, our commercial fisheries will drop. We have social benefits that come from marine protected areas, places where people can interact and learn. And we have health impacts, sometimes associated with subsistence fisheries, like in Isimangaliso. You can see the ladies collecting mussels here for their families, but also in terms of a sense of place and a sense of well-being. Those four categories are starting to seem familiar because those are exactly the things that alien species impact. So that tells us that invasions can really undermine the ability of our marine protected areas to deliver on their mandate. And that is why we really care about what happens in terms of invasions. So what do we know about marine alien species in South Africa? We've got over 100 species now, and you'll see I haven't put an exact number because we're finding them so quickly at the moment. I was working with a PhD student yesterday, and we're talking about another 3 that we think that we found. We've got a big diversity. Most of them are arthropods over here, like the European shore crab, Carcinus manus. We've got everything from algae right the way through to sea anemones. So, looking back, and before I even go that far, let's just take one step back. The foundational work that first occurred in South Africa was really around ecology. It was around marine biologists noticing a new species and going, oh, this is interesting. What ecological role is the species playing in our systems? So we have a really strong understanding of the ecology that alien species affect and their roles in South African marine systems. But in the last 20 years or so, we've really made some fundamental advancements that have moved us past just simply describing how those species function within our systems. We've become very good at detecting new alien species. We have done quite a bit of work on predicting which ones might arrive in the future. We've really got on top of how we can monitor in a way that allows us to detect those new ones, to pick them up really, really early. And okay, so you know they're here. What is it that you do? We need to actually start undertaking some management actions. So let's have a bit of a closer look at this. Whoop. Okay, and just to point out that the work I'm going to share with you now is by no means done by one person, but I'm limited with time, so I don't have the time to name each person who's been involved. And what I've done is just pop photos up in the corner of the key people that have contributed to particular sections. So thank you to all of you in advance. Right, in terms of detecting new species, here is a snapshot of some of the ones that we've found just in the last 2 years. They're really notable, they're really interesting, and if you love the marine environment, just the fact that they're there is pretty cool. So this is a long-clawed porcelain crab. It's actually really tiny. They are bright letterbox red, and that's probably why we spotted them in the first place. Really interesting species, all the way from the Med. This is a little ascidian that is about 1 centimeter tall. They're really interesting because they live together, connected by little vines, and material moves between them. So when you look at them under the microscope, you can see that material being shared. And there's questions starting to be asked about what kind of communication is happening there, what kind of sharing of nutrients, how do they work as a community. Really interesting species. One of the most interesting, though, must be this polychaete worm, Marianida. This here is the original worm. And what's happening at the back is the development of new worms. So you have an adult worm that starts to bud off new little baby worms right at the end. And so these will fall off and become new little individuals. A really good tactic if you're a marine alien species, because you're in a new place, you may not have time to have met somebody you really like yet, but you can have babies and you can make more. So this doesn't happen, this process of detecting species doesn't happen unless you're spending a lot of time in the field. A lot of the work that we do as a lab, especially recently, has been hanging over the edge of walk-ons, collecting a heck of a lot of samples, and then a lot of processing. And this photo was taken when our most recent new species was found. It's no coincidence that it's dark outside. It must have been about 8:30. One of the students Sarah realized there was something odd. They knew exactly where it had come from, and these two went trekking off in the dark, went and got some more samples, and this was them back at about 10 o'clock doing dissections. But it's because we have a solid foundation of what is present now that we're able to recognize when things are new and different. So that's resulted in 10 species that are new just since January 2025. Why is this important? It's really important because it helps us to understand the distribution of where species are along our coast, and it helps us to understand how things are changing through time. The implications of this is that we can keep SANBI up to date with our national list being spot on. As things are being recorded, it's being fed through to government immediately, as well as MPAs having a good understanding of what alien species are present either within their boundaries or just on the outside. Right, in terms of predicting what might arrive, it's so much easier to know or to define new things if you know what you're looking for. And so we've developed a process, there are a variety of ways of doing it. This is how we do it. We start with a group of species that are known to invade elsewhere. And we ask a couple of very simple questions. Can they reach South Africa, number one? So is there a pathway that they can reach South Africa? If the answer is yes, they carry on. And we end up with a group of species that could arrive. But arriving is not enough. They need to be able to survive here. So is our environment the right temperature, the right salinity to keep them alive once they're here? If the answer is yes, we know we have a list of things that can A, get here, and A, be okay when they get here. And that's called a watch list. Watch lists are incredibly helpful because you can give people who are monitoring that list. And it means that they have their minds keyed in to, I'm looking for these things, these new potential things that might arrive. We've done this for predatory crabs. This is a list of crabs on this side. You can see some of their pictures over here. And these are the 4 regions around the coast, going from the coldest Benguela region all the way to the Delagoa, which is right next to Mozambique. Where you see a tick in a white area, it means that that particular crab is perfectly suited for that area. So temperature-wise, it's A-OK. They can arrive and they can survive. Where we see the cooler colors, like over here, that area is on the cold side. Okay? Probably the crab is unlikely to survive there. When it's purple, it's definitely not. This is a warm water species. Our west coast is way too cold for it. Where we see red, it's too warm. So this is a cold water species over here. It's not going to manage in KZN. That's all fine and well if we're thinking about what our environment is like now, but we know that the environment is not static. It is changing, and one of the biggest ways that our oceans are changing is that they're warming. So by doing some modeling and looking at predictions as to what our climate will look like in the future, we can project forward and say, if we understand what those thermal requirements are of the crabs now, where will those environments be in the future? And you can see, shockingly, that most crabs are still perfectly suited for South Africa. And this is worrying because we don't have very many big predatory crabs, so any new ones that come in could really be quite devastating. You see that some of them, like this one over here, probably could have survived in Natal, but it can't anymore under future warming. And this one over here wouldn't have been able to survive in the Agulhas, but it's a little bit warmer in the future, and so there's a chance of it surviving. The take-home here, though, is that crabs are probably going to do quite well. Another example of using watch lists was when Clara Stain did one for kelp beds. So kelp beds are really important economically for South Africa, and she developed a watch list that said what species may invade our kelp forests. And Membraniptera membranacea was one of them. So you can see the kelp over here, and all of the white on it are actually tiny little bryozoans. So if we zoom in, you can see that each little bryozoan is like a little box, and when they come out to feed, you see their little feeding appendages, and they grab food out of the water. So why are we worried about Membraniptera? We worried because it's devastating to kelp beds. After heat waves, they spawn and reproduce at a much higher rate, and you can see how this kelp is totally covered with the white encrusting bryozoan, and after a while, it actually just breaks off, and you can have the whole loss of kelp beds. And considering how important our kelp forests are to us, this is really worrying. Lo and behold, what did Clara find? She found it on Muizenberg Beach. And in the car today, she was saying there's signs that it's actually in Kommetjie as well now. So at this stage, there's no massive panic because we can't pinpoint exactly where it is, but we know that it is triggered by heat waves, and we know that marine heat waves are increasing. So this is a massive concern. Okay, monitoring. We have to be actively looking for these things. And monitoring is not just myself and my students out collecting samples. Monitoring is at a government level, out there, regularly looking for things. The primary points of introduction are harbours. This is Table Bay Harbour, and right in the corner over here is Royal Cape Yacht Club, which you can see a close-up picture of here. Most of our large harbours have marinas as well. And this is important because big harbours really are the recipients of alien species. So we see them coming in here. We can have ships taking those same species and spreading them to other harbours, but we can have yachts taking them to smaller places where ships can't visit. And so we really see the spread of invasive species from these primary points of introduction. So understanding what's coming into our ports is very important. To address this, we did some experimental work in 4 harbours the whole way around the coast so that we captured all of that environmental variability. And we looked at putting out settlement plates. So these are PVC plastic plates that we hang in the water and we allow organisms to settle on. We put some of them out just like this, and others we actually put in cages. And the reason we put them in cages is because that stops predators from being able to access them and eat them. So you get the species that are hardy developing over here, and you get the ones that normally get munched by predators and are much more difficult to find, you get them doing quite well in cages. So we tested those 2 different approaches. Here you can see us putting some of those plates in the water, and this is what they look like once they're in. Once we got them back, they're just beautiful. Here you can see all sorts of organisms. These are soft-bodied ascidians. This is coming out of a cage, and that's why we've got some really lovely soft ascidians there. This is a 10 by 10 centimeter plate in my hand, and you can see the variety of different species. And everything on that plate is alien. This is a plate not from the same experiment, but one from Neisner recently, and this actually detected a brand new species unknown to the country before. That orange circle over there is a bryozoan. So what we did here, what is the science part of this? We worked at 4 different ports. We compared 2 different designs. We had these plates in for 2 different soak periods, so for different lengths of time, to test what different species we would pick up. We were really looking at trying to optimize the approach. We looked at how many do you need to put in the water, because every time you put something in the water, it costs money and it costs time because you have to have people working them. So if we're going to be suggesting that government uses a particular approach, we can't do that because it feels right. We've got to do that based on data. So all of the science went into developing data, which we then were able to present the science, so it wasn't gut feel, and we were able to present the economics that went along with that. And I'm thrilled to say that just last week, Transnet awarded the tender for monitoring of ports around our coast. And they're going to be using this methodology. What's really exciting is that we're seeing this in all of our major ports going around the coast. It's been awarded to a consultant, but that consultant wants to work with us to be able to be trained in the taxonomy. And that's just so exciting because this is our science translating into real work on the ground. And just today, we were sitting in meetings with those consultants, negotiating, chatting about how they designed their work, why we've done what we've done, and how they might be able to optimize what it is that they're going to roll out in these ports. Okay, so that's how we monitor ports, but how do we monitor just the open coast, and how do we monitor natural environments in MPAs? Well, that's how you do it. Lots of back-breaking, bending work. It's very hard work. It's labor-intensive. It is expensive. And because of those 2 things, it means that we don't monitor many places. We have these big spatial gaps, and we can't monitor very often. And so we have temporal gaps. As a result, it's really difficult to keep track of organisms spreading in the environment, and so MPAs don't have an idea of when they may be expecting new invasive species. But then, step in citizen science. Citizen science is really everybody and anybody and their phone. What you're seeing here is people taking photos of species. You can upload them to apps. iNaturalist is the big one in South Africa. It's really great because all of those records are georeferenced. You upload them. It results in a huge amount of data. It's all open access. It gives us very wide spatial and temporal coverage. So it's basically many, many eyes doing what you see Sarah and Katie doing over here. This is what an iNaturalist record looks like. This is of Carcinus manus, the European shore crab. You get all sorts of data from these, including that one was only 2 days ago. That was in 2025. It tells us exactly where the photo was taken. But there have been concerns around data quality because anyone can post a picture and anyone can call it anything. And then your auntie can say, yes, I agree with you, it's that species. And your best friend can say, yes, I agree with you, it's that species. And that's absolute nonsense. INAT's attempt to address that was something called research grade, where if you get more than 3 people saying, yes, we agree with you, then it becomes research grade. We weren't so sure, and so as part of Sarah's PhD work, we looked at the quality of data and realized that there's 3 places that things can go a little bit shady. You can have pictures uploaded that aren't clear and you can't really tell what the species is. The species itself may be a problem because some species can actually only be identified by genetics. So if you're uploading something and you're going, oh, it's the species, but we know you can't know that until you've done the genetics, that's a problem. And then also positional accuracy. We need to know that the accuracy on the phone that was taken, that took that photo, needs to be narrow enough and precise enough so that we can actually have certainty about that. If you hop onto iNat, you'll see there's a couple of terrestrial plants that are apparently growing in the middle of False Bay. Those are the records we don't want to be using. So we developed a confidence metric that allowed us to identify records that had high, medium, and low confidence. We dropped the low confidence ones and we work with the high and medium. Oh dear, jumping ahead. What that means is, instead of having 2 people out hunting for Carcinus, we can have 15,703 people out, and that generated almost 40,000 observations just on this crab. It just changes the game totally. What does this mean in terms of our MPAs? So we're looking from Longabon on our west coast around to Isimangaliso on the east coast. The number of records that we found are the height of the bars, and the numbers are telling us how many alien species we found. That doesn't look amazing. That looks like actually it could be a whole lot better, even though the numbers were quite low, excepting for Table Mountain National Park, which found 15 species and had almost 250 records. Even so, we found some really unusual and unexpected results. The sea fern hydroid over here, Paneria, was found way more west than what any scientists had known. We didn't know it had extended that far west. These two ascidians, both of which the prevailing knowledge was they are constrained to artificial habitats, they are only in harbours. They're not. They're in our MPAs, and this was the first evidence that we had for that. And then this is a maritime earwig, which was only known from KZN, and we tracked on INAT, managed to find its distribution is actually 1,500 kilometers westward. So that's a huge range extension that we never would have known about, excepting for these INAT records. So now that we know of that, we really have to start to act. So one of the ways in which we've done this was doing the first extirpation attempt in Africa of a marine species. This is the European shore crab over here. It was known to occur between Table Bay Harbour and Hout Bay Harbour. We know there had been individuals up there, but those had disappeared. And SAMBI was required to try and address removing this crab because it was listed under government legislation. What we did, because we're scientists, is firstly we trialed how do you catch these crabs? These are all ways that had been suggested in the literature, they'd been used in other places in the world. These are called crab condos, and they're really just PVC pipes which we tape together. The idea is that little crabs like to hide away, so the little ones go into the condos and you pull them up and you catch the little ones. This is a dredge which gets pulled along the bottom. The sediment goes in, goes out the mesh on the other side, but the crabs are retained. We had divers looking for crabs, and then this is the trusty old baited trap. What did we find? Well, shockingly, despite the work that was done elsewhere, not a single crab was caught in a crab condo or the dredge. And we had great faith in our divers, but they only had a 56% success rate. We hid crabs. Well, we didn't hide them. We put them down in known places on a transect, and the divers would swim them. And only 56% of those dead crabs were actually retrieved. So that meant we had really strong evidence that baited traps were what we needed to do. We tested how long they needed to be soaked for, and we needed to consider the socioeconomic context in South Africa as well, because this work was happening in Hout Bay, and these traps are awesome for catching West Coast rock lobster. So unless we had people watching them all the time, the traps were stolen. So this is a team which was put together, which ran the eradication. We needed to be able to measure the crabs. Rather than having every single crab measured, we cut holes in the lids, which were different sizes, and that allowed us to very quickly size the crabs. Before we started, we had a mathematical estimate that is how many crabs there were. This is an indication of how long it ran for. The project ran for a year, and this is the cumulative number of crabs which were caught. You can see that very quickly we caught a lot of crabs, and we were getting close to that prediction of what the population size was, and we thought we were doing really well. And then it just carried on and on and on and on. And there were just way more crabs than what we had ever anticipated. This is a graph showing the number of females. So the dots show the number of gravid females, that's females carrying eggs, also through time, and you can see how that dropped off quite quickly. The numbers above the dots are referring to the number of non-gravid females, and we're interested in gravid females because they have the potential to keep the population going. You'll see there's a very big gap over here, and that's because of an administrative issue. Our permit ran out. Our permit ran out, and even though it was a government-driven project, if you don't have a permit, you don't catch crabs. And so everything had to stop until the permits were reassessed and our new permit was issued, and then we got to carry on again. So in the end, we had to call it quits and realize we were not going to take out all the crabs in Hout Bay Harbour. This was really a very good illustration, though, of why you test these things, because then you know, you have the data. We had the data in terms of the crabs, but we also had all of the costs. So we piloted the methods, we had the evidence for why we'd chosen what we'd chosen, but we also learned a lot about the need for project administration and continuity in terms of the admin side. The science means nothing if you don't have the admin in place. And the need for secured financial support that can keep going until the end of the project was really important. Okay, so those are some of the big developments that have happened in the last while. What's the cool, exciting stuff that's kind of happening or about to happen? Firstly, we're getting really good at predicting where new detections are going to spread to. So this is the daisy anemone over here. It comes from Europe. You can see its distribution over here. Every dot is a record, and the closer the color to yellow, the more suitable that environment. It was known from Langebaan Lagoon in South Africa until we detected it in Knysna Estuary as well, so really far apart from each other. What the native range over here, what we were able to do is model and project those environments onto the South African coastline. So that's what you're seeing over here. So everywhere where it's yellow, based on where the anemone occurs in its native range, we would expect that it would occur along the South African coast. But it's no use the water being the right temperature or the right salinity. We also need the right habitat. So this is the habitat map produced by SAMBI, which is really actually very fine resolution. And it allowed us to then be able to identify places that had the right environment, but also had exactly the right habitat. Every dot on this map is a spot where the right habitat exists with the right environmental conditions. Langebaan Lagoon is black because that's where it's known from. Knysna is black because that's where it's known from. And heavens above, the grey ones, are 4 MPAs that have the right environment and the right habitat. So now we know we can warn those MPAs. This is a species that could pose a problem there. The bit that's still in development is adding movement to this model. So we need to know not only are those things in place, but how can the anemone get there? So that's the bit that's happening at the moment, the mathematical modeling that says there are yachts that move between Knysna and Gokama, or Betty's Bay is actually quite isolated, so the probability of it getting to Betty's Bay is maybe a lot lower than up to Namakwa, for instance. So that's the kind of work that we're building to be able to predict exactly where we might see these things spreading to. So this is important in terms of marine protected areas, having a bit of forewarning about which organisms might come their way. It's fine to say, be on the lookout, these species might be coming, but you need to be tracking their spread as well, so that you actually, it's not just a prediction of one day it's going to arrive. You need to know, is it getting closer? So how have we been doing that? Well, we're jumping back to an example of Carcinus here. This is the area that it was known from over there. And quite recently, we got anecdotal reports of it being up in Langebaan, as well as around Cape Point, so in that area over there. And that was actually made by Clover Mabin, who did her PhD doing the crab work. So we know that that ID was right. So we started to integrate the iNat work that we've been doing and pulled the iNaturalist records and found actually we see iNaturalist tracking it up towards Langebaan. In fact, it's even in Jacobsbaai. There's a good picture of them. We have a team that's busy resampling all of those sites. And the very, very scary thing is they're not finding it at all of the places that INAT has reported it. So we thought the scientists would be the best. We would go in and we would find it. If it was there, we were going to find it, and it would be INAT that would sometimes get it right and sometimes not. But in reality, INAT seems to be even better because it's many more eyes all of the time. Every weekend, there are lots of people out there where if we go out, we're a team of 2 or we're a team of 3, and we can spend 1 low tide checking. And so I think this has been quite a surprise for us, and I think it's going to become an even more valuable tool as we go forward. So the next step for this is actually automating the process of extracting the INAT record, running that confidence interval, and giving Sam Parks, the rangers, the ability to be at their desk and pick up, whoop, here we go. We've got this new species, and we're seeing it moving closer to Langebaan. That gives them warning. Warning for what? What are they going to do? Warning so that they can actually get some planning in place. So this is called the invasion continuum. It's really just the range from a species being absent all the way to it being established and spreading in the new country that it's arrived in. Now, in terms of management, when it's absent, what you're doing is working on prevention and preparedness. Once it's just introduced, you want to detect it really quickly. Once it's established, you really want to be containing and controlling it. But that's really at a national level. What if the species is present in the country, but it's not present yet in the MPA that you're concerned about? That really means that at this point here, you can slip out and you can undertake what's called contingency planning. And that's really about going, the species is present, we want to protect this particular environment, how are we going to do this? And that's a really important process that we've been developing. So we're going to use an example here of the warty dwarf anemone that we recently found in Knysna, and we're going to imagine that West Coast National Park is thinking in terms of contingency planning. What are the things they'd think about? Well, you'd have to look at the species and think about the species traits. What's particular about that site? What pathways connect Knysna to Langebaan? And what kind of social context is happening within Langebaan? So maybe we have traditional fishers who have access to that area. Maybe we have bird watchers who pay a lot of money to come from overseas to use that particular area. And how might global change affect all of these? So ocean warming, for instance, thermal extreme events, how does that affect this context that we're dealing with? Then we can go to the literature and we can say, okay, how have these types of things been dealt with before? Maybe the warty dwarf anemone hasn't been managed anywhere else in the world before, but maybe another anemone has. Maybe a tiny, small thing that grows, in a sedentary form has been managed. And we can develop ideas about how to manage things. It's no good to just go, oh, it worked in Canada or it worked in Australia. We need to then test those things, and by trialing them, we can trial the effectiveness, but we can also establish the costs, because maybe something is super effective, but it's flippin' expensive, and we need to know that. All of that information gets packaged into a draft contingency plan, and then we assess feasibility. And the really nice thing is that this is transparent, and it can draw in the communities that are involved, because this kind of work happening in isolation can really build barriers between the protected area and the communities which it's meant to serve. So at this point, before the species arrives, we've tested everything, we know what the costs are, we can decide on the plan. And that really means that this is evidence-based, it's transparent about how we came to our conclusions, everybody knows beforehand what their roles and responsibilities are, and finances are set aside and ready. So it's not as if a ranger finds something and then all hell breaks loose, because now how are we going to do it and where we're going to find the money? It's all set up already. So where does this leave us? Well, we're pretty good at detecting new species. We're now getting to the point where we can predict which MPAs might be at risk. That gives them forewarning to kick in the contingency planning process, and then we can leverage citizen science to track the spread of those species as they move towards those new MPAs. But the question still remains, what happens before they detect the species? This is the bit that really gets me, because I think we're doing quite well in a South African context to deal with things on the ground. But if we are not preventing new introductions, we're essentially treating the symptoms, and we really need to start working on the prevention side. So that takes us back to where we started. How do they move around? Well, in terms of mariculture, I think South Africa gets a golden tick there, because I think we're managing that pathway quite well. We have a permitting system. You can't just bring things into the country and grow them. So I think we're doing quite well there. Pet trade gets a little tick, because actually the same permitting system applies to what you import as pets. But it's very easy to order things online. At the moment, we only have, now the words have escaped me, we only have border control people who are trained in this aspect at OR Tambo, and they don't work weekends and they don't work over holidays. So if I know that, I'm sure the people who do the stuff know that. So it's very, very hard to actually manage this. So we get a little tick. Ballast water. We start with a big tick because South Africa is actually a signatory to the Ballast Water Convention, which is aiming at minimizing the use of ballast water in negative ways. But that shrinks because our Ballast Water Act has been in bill form since 2013. We have to put it into national legislation. We can't enforce anything that is just an international convention. It has to be converted into local law. It's been stuck there since 2013. So in my opinion, we actually don't have anything. So I'm going to take that tick away totally. In terms of hull fouling, at the moment, even internationally, this is something people are grappling with. The IMO is developing, they are moving towards guidelines for managing hull fouling. But in a South African context, we have absolutely nothing. So this is really where we need to be working. This is where we need to be engaging government. And it's one thing to have good work on the ground, but we really need to start to shift our thinking to be dealing with prevention. So overall, I think we've got lots of work to do. But I definitely feel hopeful and very determined that if we keep working, we can definitely make a difference. So at this point, my students are looking at the slides going, oh my word, she's committed the cardinal sin, she has a blank slide. I have a blank slide on purpose because I need to start by saying that thank yous aren't going to cut it tonight, really, but I'm going to try. Firstly, there's different teams, and the team that supports in the background, I think I have to start with Professor Charlie Griffiths. He was the very kindly prof who spoke to a very bright-eyed and bushy-haired grade 11 who was super keen, and on holiday in Cape Town, he took the time to sit down with me, and he ended up being my PhD supervisor at my grad, and it was really, he's been incredibly important in my life. Professor George Branch has been absolutely amazing. He has taught me about being a scientist, about being a mentor, about being a teacher, and just an overall great person. And I really can't imagine being where I am right now without these two very important people. And then my family, who've been absolutely amazing. I am so grateful, Mum and Dad. Brad and Meg, I know you're online. I hope you can hear me. I think the only time I ever heard a hard no was when I was banned from dissecting seafood on my dinner plate. Other than that, there's been nothing that you guys haven't supported. You've done field work with me in the middle of the night, up the north, up the coast. It is incredible because you believe in me, I believe in me. And I really thank you so much. And then because we're in Stellenbosch, I can say that I'm moving from my familie to my gesin. That was one of the very confusing things for me as a child, learning Afrikaans. Damien and Kira and Vin, I could not do what I do without you. I am deeply appreciative of each one of you. You're a huge part of this, and I did resist putting photos up of when you were little and you were doing field work, so you owe me. And then to the team that is actually at the forefront, I have had the most amazing group of students. You guys inspire me, you keep me on my toes in so many ways, but it keeps life exciting, it keeps my work exciting, and I'm very, very appreciative of having been able to shepherd you through this interesting time in your lives and to be able to share all of that with you. So thank you, every single one of you. And then thank you to everybody who's here tonight. Thank you for shifting your calendars to be here, because we were meant to be here 48 hours ago. And thank you to everybody online for taking the time to spend this with us. Thank you.
Thank you very much, Tammy. I think we all agree that this is fantastic work. We are so, so proud of you. Thank you for making Stellenbosch your home, and we hope that you will continue working in this wonderful field for many, many long years to come. And we would want to see a lot of PhD students and those of the students here. Congratulations for working with your professor, and thank you for building her CV. Someone agrees. The floor is now open for a few questions. Anyone from the audience or online? I'm not sure if we can hear questions from on. Oh, just from here. Okay, so we'll only be taking questions from this venue here. Any questions? At the back.
Hello, Tammy, and thank you for your lecture. I wanted to check in with you if you could help us. What has enabled South Africa's growing leadership in this marine invasion space, and what do you think needs to happen to strengthen collaboration and also capacity across the broader African continent?
Sure. That has to be the CIB. The Centre for Invasion Biology has really driven all of this. Without the CIB's faith and funding and backing, none of this work could have happened. And they have drawn the students that I've supervised, they've drawn to Stellenbosch. And it's very exciting to see the CIB starting to develop an African interest. And the plan over the next few years is to spread into Africa. And we are already doing that in a marine context. I have a PhD student who's based in Namibia. I think Kathleen's probably online. And so we're taking those steps because invasions are not a problem which are confined to political boundaries. And so we need to start working with our neighboring countries and spreading this work through Africa.
Anyone else?
Hi, Tammy. I just have a quick question. All the food you guys caught in holiday, what happened to that?
Which food?
The crab? Oh, sorry. All the crabs and the rock lobsters you guys caught in Hout Bay. Are we invited for a seafood broil? What's going on?
Oh, goodness. My PhD supervisor used to have dinner evenings where we all had to bring what we were working on. So sometimes marine scientists get to eat their food, but no, we didn't eat any of those because the harbors are so laden with heavy metals that the crabs were laden with heavy metals. We had a lot of people asking if they could be used in that way, but they can't. And there was even, we looked at using them as fertilizer, but the heavy metal levels were too high there as well. So it seems like such a waste, yeah.
I can perhaps just end off by asking, so are there any feel-good stories around the world? Because it seems to me like a daunting task to, it's one thing to detect, to find them and to determine where they are, but the world, not only in South Africa, is there any feel-good story to say that it was found, removed successfully and kept out forever?
So I was going to say yes until you said kept out forever. Because yes, we have had good responses, primarily in Australia. They take no prisoners. There is an example of them detecting something in a marina. They actually shut it off with a lock, and they blitzed that entire thing, even though it was a long weekend. It was the organism was removed by the following week. But the problem is the reintroduction. And until we start to deal with that at a global scale, no one country can do it on their own. So we have to get the Ballast Water Act, we have to get our Ballast Water Act in, but all countries have to get a handle on ballast water so that we stop sharing things. But there are feel-good stories, sometimes not in terms of detection, but one of the feel-good stories in a South African context is the Mediterranean mussel, which is so dominant on our coast, has had some negative effects, but it's had one unanticipated amazing effect, and that's that oyster catchers were endangered along our coast. And they were endangered primarily because of habitat disturbance, but they started producing triplets, little chicks that were in threes rather than one at a time. And the reason that was happening was because they were able to eat so much more because there were so many more mussels. So they did better, they had more chicks, and now they're actually not endangered anymore. And so we have to look at invasions holistically. It's not all doom and gloom. Yeah.
Oh, that's a fantastic story. Thank you so much. Last go, any final questions? If not, I would like to introduce someone from the Rectorate, Professor Richard Stevens. He will just do a final handover of the certificate and also have some comments.
Dear Professor Robinson-Smythe, family, colleagues, friends, godparents of Prof. Smythe, thank you for delivering an engaging inaugural lecture, and congratulations on this achievement, which is a very significant event in the university calendar. An inaugural lecture serves as a platform for the Faculty of Science and Stellenbosch University to acknowledge the esteemed status and expertise of a senior academic colleague. It also provides an opportunity for scholars to showcase their academic trajectory over time. The rarity of sharing a professional milestone with an audience that includes family, friends, students, and colleagues adds to this significance. Furthermore, an inaugural lecture facilitates the dissemination of contemporary topics. Prof. Tammy illuminated a discipline of maritime protection areas and conservation that is highly relevant today. I mentioned before the lecture start, I made a note to self the whole day not to say conversation, but to say conservation. And I actually managed that. If I may digress a little bit, I think often of my favorite song, Kashmir, by Led Zeppelin. The song refers to the metaphorical journey through life, through wastelands and extreme beauty. Kashmir, despite its title, is not about the region in India, between India and Pakistan. It's a metaphorical journey of spiritual exploration and transcendence, inspired by a long drive through the Moroccan desert by Robert Plant. The lyrics evoke a sense of timelessness, vastness, and the search for something greater than oneself, and illustrated tonight by the vastness of the ocean. In essence, Kashmir is a powerful and evocative song that uses a geographical reference as a starting point for broader exploration of the human experience and the quest for meaning. Sometimes academia feels like these highs and lows, and Prof. Robinson-Smythe not only illustrated the beauty of her journey, but also that maritime journeys have huge impact, not necessarily positive for conservation. Now I'm entering into dangerous ground, a lawyer trying to summarize a science lecture for key points, so I'll try my best. Firstly, why marine invasions matter. Alien species spread globally, often through shipping, recreational vessels, and mariculture, and can harm native biodiversity as well as human health and well-being, as well as cause economic harm. 2, the importance of maritime protected areas and conserving them. 3, the need for evidence-based prevention and management. Achieving conservation goals cost-effectively requires understanding how species arrive and spread, and ensuring that interventions are guided by solid scientific evidence. 4, the growth of South African marine invasion research. Over the past 20 years, understanding of marine invasions in South Africa has expanded rapidly, leading to key achievements in the field. Example that Ashley illustrated tonight: detection, predicting, and monitoring. 5, current understanding of coastal invasions in South Africa. The lecture reviews what is currently known about invasive species affect South African coast and the invasion dynamics there. Then lastly, future-facing developments and leadership in Africa. It highlights recent exciting developments positioning South Africa at the forefront of addressing marine invasions. It was quite ironic, though, that she said Australia is at the forefront of this because they take no prisoners, having been prisoners that they went there in the first place. But that's besides the point. So, with such insight, knowledge, and dedication to the discipline, the ground is fertile for further research, and it's reassuring to see her students here tonight. I think as academics, we have the privilege of what has been termed, quote, the sublime art of investigating facts and occurrences, of discerning truth from falsehood, like uploading false photos and fake news, and certainty from doubt, unquote, in order for our research to strengthen, straighten, and extend the mind. On behalf of the SU community, I extend my best wishes to you, Prof. Robinson-Smythe, for the practice of this sublime art and the successful continuation of your academic journey. And with that said, it is now my pleasure to hand over the certificate to Prof. Robinson-Smythe.
Thank you so much, Professor Stevens, and you did a sublime job. I would like now to, for the last bit of this wonderful occasion, to call upon Prof. Guy Midgley. He will do the vote of thanks and close us for us. And then afterwards, you are welcome to join us for something to eat and to drink.
Thank you. Well, it's a great privilege to be able to have the last word and to close off on what an amazing event. I think we've had a real treat, Tammy. Thank you. So first, yes, I want to thank you for a brilliant talk, a marvelous journey through your amazing work, which links fundamental science all the way through to policy and impact. That is something truly special. I've had the pleasure of managing Tammy for the last few years, although I have to correct that. She's a very, very easy person to manage, probably because she knows how to manage me very well. And that has happened because Tammy has the chair for protected areas and invasive species in the Center for Invasion Biology, which is in the School for Climate Studies. And I also want to thank you, Tammy, for your vision in incorporating climate change into your invasions work. Yeah, I mean, you're really good at a field. It's yeah, it's sometimes a stretch to bring in an additional element to your work, but you've done incredibly well in bringing in this additional aspect, and I think you've done some really groundbreaking work there. But Tammy, I'm not going to reflect on all the content of the talk, but just talk a little bit, just briefly, to thank you for your work in building up the chair. It's not easy to build up a new thing, especially when there are lots of existing things that are sometimes in the way. So you've done an incredible job, and the first 3 years of funding was indeed extended because Tammy has been so successful in driving her chair. And so she got an additional 3 years of funding. And you can see the result of this investment in her students and in the work that's been done and in the impact, in the societal impact. I think it's amazing what you've achieved. Of course, off the base of all your original work, but also leveraged through the money that has come into the chair from a very generous funder. So thanks for the wonderful talk. Thanks for your passion. I think you can see how Tammy's passion rubs off on her students, her brilliant team, the large number of students that she's attracted, people who are very dedicated to the team. It's a real testament. And it's not easy. I know, you're a mom, you're a partner. You've got lots of pressures to balance, and you've done an amazing job on that. So, and you've added a lot to the credibility of Stellenbosch University and to the Botany and Zoology Department, Science Faculty, and the CIB, and the school. So more broadly, thank you everybody for coming. I think we've all enjoyed it thoroughly, but it's wonderful to see so many people here. I want to briefly thank the organizers, JD, Jonathan, Hazel, and Busa from the Faculty of Science for doing all the organizing, the team from the registrars, RC, and others who were involved in organizing this event. Rectorate and university leadership, who we must thank for their continued support of all of our scholarship and creating the environment in which people can achieve these sorts of things. And I think you can see the payback on that investment. So once again, congratulations, Tammy, and I will release you all. But before you go, I have been instructed to say that you all need to stand to allow the procession to proceed. Thank you.